Hello everyone, my name is Marina Idail. This time around, we will go over the practice questions. Now heads up lang po, babagalan ko with each question so you can keep up. Now I will read the question twice. Also later on, you will notice the questions are colored black, but some words are in red. Those highlighted in red will give you a hint or a clue of the answer. So gear up as we try to recall some concepts in clinical lab science. So if you're ready, let's begin. Number 1. Which disease might be indicated by antibodies to smooth muscle? Again, which disease might be indicated by antibodies to smooth muscle? A. Atrophic gastritis, B. Chronic active hepatitis, C. Myasthenia gravis, or D. Sjogren's syndrome. Again, which disease might be indicated by antibodies to smooth muscle? Okay, the correct answer is letter B, chronic active hepatitis. Remember, antibodies to smooth muscle are found in the serum of up to 70% of patients with active chronic hepatitis. Just remember asthma, anti-smooth muscle antibody for chronic active hepatitis. Now for Graves' disease, so we have the antibodies and their disease association. For Graves' disease, we have anti-TSH receptor antibody or anti-TSHR. For Hashimoto's thyroiditis, we have anti-microsomal antibody and anti-thyroglobulin antibody. For myasthenia gravis, we have anti-acetylcholine receptor antibody. For Goodpasture syndrome, keyword is basement membrane, we have the anti-glomerular basement membrane antibody. And for chronic active hepatitis, again, we have anti-smooth muscle. Another, we also have rheumatoid arthritis. We have anti-IgG, or we call that as rheumatoid factor. For CREST syndrome, we have anti-centromere antibody. Now for pernicious anemia, we have the anti-intrinsic factor antibody. For SLE, we have the anti-nuclear antibody, or we call that as ANA, and anti-double-stranded DNA and anti-Smith. For vaginous granulomatosis, remember ANCA, anti-neutrophilic cytoplasmic antibody. Now for primary biliary cirrhosis, we have the anti-mitochondrial antibody. For multiple sclerosis, we have the anti-myelin antibody, and for type 1 DM, we have anti-insulin, anti-beta cells, and anti-glutamic acid decarboxylase. Also, we have for Sjogren's syndrome, anti-SSA and anti-SSB. For celiac disease, we have anti-tissue transglutaminase or anti-gliadin, and also for rheumatoid arthritis, anti-CCP or anti-citrulinated peptide. Next, number 2, an RPR card test performed on CSF was non-reactive. The physician was skeptical and asked for a repeat test. Now, the RPR result was reactive, 1:1 dilution. The result, take note, RPR card test was used on CSF. Choice letter A should be reported as non-reactive. B should be reported as reactive, 1:1 dilution. C is non-conclusive and should be repeated on a new CSF sample. Or letter D is unreportable. The RPR test should not be performed on CSF. The correct answer is letter D. Is unreportable. The RPR test should not be performed on CSF. Now, syphilis is a sexually transmitted infection caused by Treponema pallidum. In the diagnosis of syphilis, serological tests play an important role. So, of course, the specimen na gagamitin natin is serum. Now, serological tests are useful in the latent stage of the disease dahil mahirap i-sustain sa culture ang treponemes. And also, yung lesions are usually absent. Now, under serological testing, there are two. We have the non-treponemal test and the treponemal test. Non-treponemal tests are done to determine disease activity. They are mainly used as screening tests. Under non-treponemal tests or screening tests, we have RPR or the Rapid Plasma Reagin in VDRL or Venereal Disease Research Laboratory. Now, under treponemal tests, treponemal tests are used for disease confirmation. Examples, we have FTA-ABS, or the fluorescent treponemal antibody absorption, and for THPA, treponema pallidum hemagglutination absorption. Now, VDRL tests may also be performed on CSF samples. It has long been considered as the best serological test for the diagnosis of neurosyphilis. Again, VDRL is the gold standard for the diagnosis of neurosyphilis. Also have the rapid plasma reagin. This test is easier to perform, but its appropriateness for use on CSF is controversial. Likewise, it cannot replace the VDRL for diagnosis. Although may problema sa sensitivity ni CSF VDRL, remember the CSF RPR has a high false negative rate. So the correct answer for that question is unreportable. RPR test should not be performed on CSF because it is negatives. Number 3, what is the titer in tube number 8 if tube number 1 is undiluted and dilutions are doubled? Again, what is the titer in tube number 8 if tube number 1 is undiluted and dilutions are doubled? A, 64, B, 128, C, 256, or D, 512. Okay, correct answer is letter B, 100. Let's go to titer. Titer is a way of expressing concentration, so it employs serial dilution to obtain approximate quantities from a procedure that evaluates as positive or negative. Now, the titer corresponds to the highest dilution factor that still yields a positive reading. Again, highest dilution factor that yields a positive reading. Now, in the given question, tube 1 is said to be undiluted. That means we are not going to include tube 1. So we go on to tube number 2, 3, 4, 5, 6, 7, 8, and so on. Also in the question, dilutions are doubled. That means the dilution is 1:2 or 1/2 for every tube. Now we were asked to look for the titer of tube number 8. So simply we're just going to multiply all the dilutions from tube number 2 down to tube number 8. The answer is 1:128 or 1/28 or a titer of 128. Number 4, the H antigen is found in lowest concentration on what phenotype of red blood cell? Again, the H antigen is found in lowest concentration on what phenotype of red blood cell? Pa-highlight naman po ng H antigen and the lowest concentration. Choices. Okay, letter A, we have group B, choice B, group A1B, letter C, group A2B, or D, group O. Okay, correct answer is letter B, group A1B. Now, for A and B antigen synthesis to occur, a precursor called the H antigen must be present. So in the RBCs, the enzyme that synthesizes the H antigen is encoded by the H locus or FUT1. In saliva and other bodily secretions, the enzyme that synthesizes the H antigen is enclosed by the SE locus. Remember, blood group na may highest amount of H antigen is group O, followed by group A2, then group B, and then A2B. And yung may least amount of H antigen ay group A1B. Okay, number 5, which of the following could be the possible cause of the ABO discrepancy? Again, which of the following could be the possible cause of the given ABO discrepancy? So as you can see, there were no reactions in both the forward and reverse grouping. In the forward grouping, the patient types as an O. Now, if the forward grouping is O, it should yield a reaction to both A1 cells and B cells in the reverse grouping. But instead, walang reactions. This means that there are weakly reacting or missing antibodies. So choices, is it A, acquired B phenomenon, B, subgroup of A or B, C, immunodeficient patient, or D, polyagglutination? Correct answer is letter C, immunodeficient patient. Now just a recap, ABO when unexpected reactions occur in the forward and reverse grouping. May be caused po ito again by problems with the patient's serum as seen in the reverse grouping, or problems with the patient's red cells as seen in the forward grouping, or problems with both the serum and cells. Now all ABO discrepancies must be resolved prior to reporting a patient or donor ABO group. So let's discuss Group 1 ABO discrepancies. Now Group 1 discrepancies are associated with unexpected reactions sa reverse grouping po due to weakly reacting or missing antibodies. Again, kapag ang reaction in the reverse grouping is weak or missing, a Group 1 discrepancy should be suspected because normally, RBC and reverse grouping reactions are very strong. Isang reason po kung bakit may missing or weak isoagglutinins ay dahil yung patient ay may depressed antibody production, or in other words, di siya nakaka-produce ng ABO antibodies. Now, common populations with discrepancies in this group include newborns. Remember, the production of ABO antibodies is not detectable until 4 to 6 months of age. Also, for elderly patients, where the production of ABO antibodies is depressed, and also patients with leukemia. Example, chronic lymphocytic leukemia or lymphoma, demonstrating hypogammaglobulinemia. And we also have for patients using immunosuppressive drugs that yield hypogammaglobulinemia. Patients with congenital diseases or acquired agammaglobulinemia. We've also patients with bone marrow or stem cell transplantations. Usually patients develop this hypogammaglobulinemia from therapy, and they start producing a different population from that of the bone marrow. And also, ABO subgroup. In patients whose existing ABO antibodies may have been diluted by plasma transfusion or exchange transfusion. Now for Group 1, ABO discrepancy, how do we resolve this problem? Again, how do we resolve Group 1 ABO discrepancy? Simply, we check the patient's history. Now, if you encounter this problem, you might consider the age of the patient. So the sample might be from a newborn. Remember, newborns would not have ABO antibodies in the serum until the age of 4 to 6 months. Now, if the history naman indicates an elderly individual or the diagnosis indicates hypogammaglobulinemia or for immunosuppressed patients, then the best way to resolve this discrepancy is to enhance the weak or missing reaction in the serum. Paano po ba ine-enhance yung weak or missing reaction in the serum? This is usually performed by incubating the patient's serum with reagent A1 and B cells at room temperature for 15 minutes. Or pwede rin po by adding 1 or 2 drops more plasma or serum. Now, kapag wala pa ring reaction after centrifugation, the serum cell mixtures can be incubated po at 4 degrees Celsius for 15 minutes. Before we go to number 6, let's take a look at the A subgroups. So in 1911, von Dungern described 2 different A antigens based on the reactions between group A RBCs and anti-A, and between group A RBCs and anti-A1. Now, group A RBCs that react with both anti-A and anti-A1 are classified po as A1, whereas those that react with anti-A and not anti-A1 are classified as A2. Now, later studies have found that the number of A antigens is much higher than A1 RBCs than A2 RBCs. Again, A and A1 antigens for A1 subgroup and A antigen only for subgroup A2. And it was also shown that A1 RBCs possess A antigens on the branched and unbranched core structures, whereas yung sa A2 subgroup naman predominantly po sa unbranched structures. But take note also, A1 subgroup does not produce a naturally occurring anti-A1. Again, for subgroup A1, no anti-A1. A2, on the other hand, may produce anti-A1. Now for number 6, which of the following could be the possible cause of the given ABO discrepancy? Again, which of the following could be the possible cause of the given ABO discrepancy? A. Acquired B phenomenon, B. A1 subgroup, C. A2 subgroup, or D. Polyagglutination. The correct answer is letter B, A1 subgroup. So sa tanong na to, we see that in the forward typing, the sample is definitely blood type A, since it strongly reacted with anti-A, and with reaction sa B cells in the reverse typing. So we have to choose between choices B or C, tama, subgroup A1 or subgroup A2. But notice that there was no reaction with the A1 cells. So no anti-A1 was produced. So that's true for A1 subgroup. At the same time, you might also consider its reaction with the O cells. 3+, right? So this is probably subgroup A1 with a potent anti-H. By the way, acquired B phenomenon naman, okay, bakit hindi po pwede acquired B phenomenon? Acquired B phenomenon occurs when RBCs from blood group A patients come into contact with gram-negative bacteria. What happens is that the bacteria carrying an enzyme removes a part of the A antigen, leaving behind a modified sugar na nagre-resemble po ng B antigen. So pag acquired B phenomenon, blood type po talaga ng pasyente ay A, pero you will expect that there will be a reaction in anti-B. For number 6, again, we have letter B. Number 7, which of the following could be the possible cause of the ABO discrepancy? Again, which of the following could be the possible cause of the ABO discrepancy? A. Acquired B phenomenon, B. A1 subgroup, C. A2 subgroup, or D. Polyagglutination. Again, which of the following could be the possible cause of the ABO discrepancy? Correct answer is letter C. A2 subgroup. Now, since it reacted with anti-A, in the forward grouping, we have to presume that this is an A blood group. But notice the reaction in A1 cells. So it produced anti-A1. So this is A2 subgroup. Again, with anti-A1, that's subgroup. For number 8, which of the following is most helpful to confirm a weak ABO subgroup? Again, which of the following is most helpful to confirm a weak ABO subgroup? A. Adsorption elution, B. Testing with A1 lectin, C. Neutralization, or letter D. Use of anti-A1B. Again, pa-highlight naman po ng weak ABO subgroup. Correct answer is letter A. Adsorption elution. Now, adsorption and elution techniques are used to detect ABO antigens that are not detectable by direct agglutination. When we talk of adsorption, it is the removal of antibody from the serum. And this is done by combining a serum sample with RBCs under optimal conditions. Now, after the antibody attaches to the membrane-bound antigens, and the serum and the cells are separated, may iwan na lang po yung specific antibody na naka-attach sa red cells. So it may be possible to harvest the bound antibody by elution. Now, the cells are incubated with the antibody to the antigen expected on the RBCs. So an elution method is performed and the antibody in the eluate is tested for recovering anti-A or pwede ring anti-B. So depende po yan sa specificity na ginamit sa adsorption. Again, weak ABO subgroup of adsorption elution. Number 8 letter. Now for number 9, the results of D-typing on a patient using a high protein anti-D reagents are. Again, the results of D-typing on a patient using a high protein anti-D reagents. As you can see, patient cells are made to react with anti-D and RH controlled. So it reacted sa AHG phase. Now, which of the following is the correct interpretation of these results? Again, it reacted to AHG phase. Is it A, Rh negative, weak D positive, B, Rh positive, weak D positive, C, Rh negative, weak D negative, or D, invalid Rh typing? Now, correct answer is invalid Rh typing. When Rh typing or D typing is performed using a high protein reagent, again, using a high protein reagent, a high protein control reagent is also reacted with the cells, with a separate aliquot of the cells. This high protein control lacks anti-D. So it is expected to give a negative reaction. Again, it is expected to give a negative reaction. Yung reaction po ng Rh control serum with the cells na nakikita nyo is not due to anti-D reacting with the D antigen on the cells, but this is actually due to an interaction of the high protein matrix of the anti-D reagent. So therefore, yung positive reaction na to with the Rh control serum prevents the interpretation of the presence or absence of D antigen. So we cannot say or we cannot tell if the patient is Rh positive or negative, or weak D positive or weak D negative. So the answer would have to be invalid Rh or D typing. The summary of Rh typing result using high protein anti-sera. Rh positive individuals are expected to give positive results with anti-D, negative with Rh control. For Rh negative patients, they are expected to give negative results with anti-D and negative results also with Rh control. And for invalid, Rh typing may be positive or negative with anti-D and positive with Rh control. Next, for number 10, glycophorin A and glycophorin B possess antigen sites for which blood group system? Again, glycophorin A and glycophorin B possess antigen sites for which blood group system? A, Duffy, B, Kidd, C, Lewis, or D, MNS? Correct answer is letter D, MNS. Remember, the M and N antigens reside on glycophorin A, while SS, capital S and small s, and U antigens reside on glycophorin. Characteristics of M and S antigens. M frequency is roughly equals N. So each of them 75% po. And we have small s is more frequent than capital S. Around 90% of the population for small s, for capital S, 50% for Caucasians, and 30% for African Americans. So if the patient or if the individual is S minus, he or she may also be U negative. So this S minus is seen in 2% of African Americans. Remember that U is extremely high frequency, and we also have the Muir antigen. The Muir hybrid antigen is seen in nearly 10% of Chinese. Also, Vishya graminea lectin reacts against N antigens, bodies. So significant antibodies can form, more frequent in some areas than anything, but anti-A or anti-B. Now, anti-M and anti-N can usually be ignored unless they are reactive at 37 degrees Celsius. And effect varies by enzyme, but enzymes generally decrease all M and S antigens except U. So for M and N antibodies, they are mostly opposite of S, and U antibodies. Now, characteristics for anti-M and anti-N, they are naturally occurring. Unlike anti-S and U, they require exposure. Anti-M and anti-N, nature, they are of the IgM type. So they are called cold antibodies. And anti-S and anti-U, they are of IgG in nature, so warm antibody. Anti-M and anti-N show dosage, and anti-S and anti-U show minimal dosage. And anti-M and anti-N are clinically insignificant, whereas anti-S and anti-U are significant and clinically significant antibodies. Next, for number 11, all of the following exhibit dosage except, again, all of the following exhibit dosage effect except So when we say dosage effect, stronger agglutination when a red cell antigen is expressed from homozygous genes. So choice letter A, is it Duffy, B, Kidd, C, Lewis, or D, M&S? Again, all of the following exhibit dosage effect except answer is letter A. Now remember, those enhanced by enzymes. What blood groups are enhanced by enzymes? Remember, the mnemonic clipper. Just remember the mnemonic clipper. Clipper, we have Kidd, Lewis, I, P, P1, and RH. Again, clipper, Kidd, Lewis, I, P1, and RH. Those destroyed by enzymes include Duffy and M&S. And those showing dosage effect, just remember the mnemonic, Duffy the rich kid in Manila. Again, Duffy the rich kid in Manila. So we have Duffy, RH, Kidd, M&S, and Lutheran. Next, for number 12, an antiglobulin cross-match is performed with a donor red blood cell unit. The antiglobulin cross-match result is a 2+ agglutination reaction. What is the most likely explanation for this result? Again, an antiglobulin cross-match is performed with a donor RBC unit. Now, the antiglobulin cross-match result is a 2+ agglutination reaction. Choices: A. Recipients RBC are demonstrating polyagglutination. B. Recipients RBCs have a low frequency antigen. C. Recipient possesses an IgG alloantibody. Or letter D. Recipient possesses a cold autoantibody. Okay, so the correct answer is letter C. Again, for this agglutination to occur, an additional antibody which reacts to the Fc portion of the antibody or the component of the complement must be added to the system. So this will serve as a link or bridge between the antibodies or complement, coating the red cells, causing this agglutination. So given may 2+ agglutination on the antiglobulin, take note that means there is an antibody coating the red cells. Now, agglutination with polyspecific ASG indicates that either IgG antibodies or complement are coating the recipient. Next, for number 13, which of the following is not a step in platelet production? Then which of the following is not a step in platelet production? A. Soft spin to sediment red blood cells. B. Hard spin to separate platelets from platelet-rich plasma. C. Resting phase at 1 to 6 degrees Celsius. Or D. Resting phase at 20 to 24 degrees Celsius. Correct answer is letter C. Remember, platelets are not supposed to be refrigerated. They should be maintained always at room temperature. Now let's go to the basic blood component preparation. Now going to the basic blood component preparation, whole blood is subjected to light spin or soft spin to sediment RBCs, thereby separating the platelet-rich plasma. This platelet-rich plasma is then subjected to hard spin para makaproduce tayo ng platelet concentrate. From the same spun component, platelet-poor plasma is separated and when subjected to -18 degrees Celsius, forms fresh frozen plasma. Or a shorter way of producing FFP is by separating whole blood to hard spin, separating RBCs from plasma, and freezing the plasma to -18 degrees Celsius will produce FFP. Number 14, a blood supplier ships 3 units of pooled cryoprecipitate. Each pool consists of 5 units of cryoprecipitate. If 1 unit is thawed at 5 p.m., when must it be dispensed from the blood bank? Again, a blood supplier ships 3 units of pooled cryoprecipitate. Each pool consists of 5 units of cryoprecipitate. If 1 unit is thawed at 5 p.m., when must it be dispensed from the blood bank? Okay, take note, pooled cryoprecipitate po ito. Choices letter A, before 9 p.m. Choice letter B, before 11 p.m. Choice letter C, before 12 a.m. Or choice letter D, before 5 p.m. on the next day. Correct answer is letter A, before 9 p.m. Now, cryoprecipitate is prepared from plasma and it contains fibrinogen factor 8 what else? VWF and fibronectin, also factor 13. It is stored, remember, frozen, and it must be transfused within 6 hours of thawing or 4 hours of cooling. Now going to the other blood components and their major indications. So of course, whole blood is used for symptomatic anemia with large volume deficit. Also take note, deglycerolized or washed RBCs for severe allergic reactions. Leukoreduced or leukocyte-reduced RBCs are used for febrile reactions. And we also have pooled platelets, okay, bleeding due to thrombocytopenia or platelet function abnormality. Leukoreduced again for prevention of febrile reactions. Granulocyte spheresis for neutropenia with infection unresponsive to appropriate antibiotics. Now, pooled cryoprecipitate again for factor 13 deficiency and hypofibrillation. Number 15, intraoperative autologous blood stored at 1 to 6 degrees Celsius should be administered within how many hours? Again, intraoperative autologous blood stored at 1 to 6 degrees Celsius should be administered within A, 4 hours, B, 6 hours, or C, 12 hours, or D, 24 hours. Again, pa-highlight naman po ng intraoperative autologous donation at yung 1 to 6 degrees Celsius po. Correct answer is letter B. Sa intraoperative autologous donation po, okay, the blood may be stored at room temp for up to 6 hours. Again, room temp for up to 6 hours or at 1 to 6 degrees Celsius din po for up to 24 hours. As long as the second temperature has begun within 4 hours from the end of collection. Again, pag room temp po, up to 6 hours. And pag ref temp, pag 1 to 6 degrees Celsius po for up to 24 hours. Again, pag ref hours. Next, for number 16, antibody screen and antibody identification cells are grouped. Again, highlight naman po ng antibody screen, antibody identification. A, A1 and B, B, A and O, C, O only, D, AB only, or E, which I haven't included there, any ABO group. Again, antibody screen and antibody identification cells are grouped. Okay, correct answer is letter C, O only. Now, antibody identification cells are also human products po. As with the antibody screen cells, these are group O cells, okay, that have been tested for the presence of most commonly encountered antigens. So remember, screening cells are used for antibody detection, and panel cells naman po are used for antibody identification. Now for number 17, group O Rh positive cells are used for antibody screening tests because, again, group O Rh positive, pa-highlight naman po. A, anti-A and anti-B do not react with O cells. B, anti-A1 is detected using O cells. C, most recipients are O and Rh positive. Or D, weak A or B subgroups react with O cells. Anong answer natin? Correct answer is letter A po. Anti-A and anti-B do not react with O cells. Now, group O cells are selected to avoid interference from ABO system antibodies. And these cells are also selected to have as many antigens corresponding to common antibodies as possible para, okay, to allow detection of unexpected clinically significant antibodies. Ginagamit naman po natin yung Rh positive cells para most likely maka-expect po tayo ng detection of unexpected anti-B. Choice B po ay wrong. Again, mali po yung choice B. Why? Because anti-A1 is not detected using O cells. Rather, it is detected with A1 cells. For number 17. Now, for antibody screen, just a run-through or quick run-through of the interpretation of antibody screen. Just in case nakalimutan po ninyo. Agglutination or hemolysis at any stage of testing is a positive test result. Pag may positive result, that's the time we proceed sa antibody identification studies. Now, kung mag-i-investigate po tayo, we should consider the following questions po. Number 1, in what phase did the reactions occur? Again, in what phase did the reactions occur? Now, antibodies po of the IgM class react best at room tempo or lower. They are also capable of causing agglutination. So they will react at the immediate spin phase. Again, antibodies of the IgM class react at the immediate spin phase po. They react best at room temp, so may reaction po sa immediate spin. Now, antibodies naman of the IgG class react best at the AHG phase. Again, pag antibodies po, yung class niya po is IgG, it will react with the AHG phase. Another thing that we have to consider is the autologous control negative or positive. Although di po natin makikita dito because some technologists choose to omit the autocontrol when performing the antibody screen dahil na rin sa complexity nito. So sometimes dinadagdag lang po nila to pag nagpe-perform ng antibody identification studies. Now remember, a positive autologous control may indicate the presence of auto-antibodies po. Again, pag positive po yung autocontrol, it may indicate the presence of auto-antibodies or antibodies to medications. Or if the patient has been recently transfused, the positive autologous control may be caused po by alloantibody coating the donor RBCs. Now, pag a positive antibody screen naman po, tapos negative autocontrol, that indicates an alloantibody has been detected. Another thing, for number 3, did more than one screen cell sample react? Again, if so, did they react at the same strength and phase? So, a single antibody specificity should be suspected po pag yung screen cells na may positive reaction ay nagre-react at the same phase and same strength. Again, single antibody specificity po pag nag-react at the same phase and same strength. Now, multiple antibodies are most likely naman po pag ang screen cells ay nagre-react at different phases or strengths. For example, and again, multiple specificities po pag nagre-react at different phases or strengths. Also, another thing to consider is hemolysis. Is it hemolysis or mixed field agglutination? So, antibodies such as anti-Lewis A, anti-Lewis B, anti-PP1-PK, anti-VEL are known to cause in vitro hemolysis po. Mixed field naman po is usually associated with anti-SDA and Lutheran antibodies. Also, are the cells truly agglutinated or a RULU present? Now, serum po from patients with altered AG ratios, for example po sa mga may multiple myeloma or those who have received plasma expanders, that may cause non-specific aggregation of RBCs are known as RULU, known to cause RULU po. So RULU, remember, is not a significant finding in antibody screening test, but sometimes na-confuse po yan with antibody-mediated agglutination. For number 18, a DAT result is as follows. Polyspecific AHG is equals to 1 plus, anti-IgG is negative, check cells po 3 plus. The most etiology of this positive DAT is, again, given the following DAT result. Polyspecific AHG 1 plus, anti-IgG is negative. Now, the most etiology of this positive DAT is choices. Letter A, anti-IgG, B, anti-C3, or C, anti-I, or letter D, anti-A1 in an A1 individual. Okay, correct answer is letter B, anti-C3. Now, just a recap. There are two types of antiglobulin tests. We have DAT and IAP. DAT, direct antiglobulin test, and the indirect antiglobulin test. Now, DAT detects red cells coated in vivo with immunoglobulin or complement. Now, remember, washed red cells from a patient or donor are tested directly with anti-human globulin reagents, forming a visual red cell agglutination. Now, this test is used to detect hemolytic transfusion reaction, HDN, or autoimmune hemolytic anemia. Now again, for this agglutination to occur, an additional antibody which reacts to the Fc portion of the IgG antibody or with a component of complement must be added to the system. And this will serve as a link or bridge between the antibodies or complement coating the red cells, again causing agglutination. So AHG reagent, we have the polyspecific and we also have the monospecific. Polyspecific anti-human globulin is a blend of anti-IgG, anti-C3b, and anti-C3d. Monospecific reagents, anti-IgG alone or anti-C3b, anti-C3d alone. Noted that may be set up only with the polyspecific tube first po, and then repeated with the monospecific reagents if the polyspecific test is positive. So ginagawa po to, okay, yung inuuna yung polyspecific kaysa sa monospecific if generally done when you have no indication of what the result of the test will be. However, if you're expecting the DAT to be positive, it is advisable to set up tubes for polyspecific and monospecific reagents at the same time and test them all together. So no agglutination indicates a negative DAT. That means nothing is coating the patient cells in vivo. Then agglutination with polyspecific AHG indicates a positive DAT. That may be due to either IgG antibodies or complement. Now, to determine specifically if it is an antibody or complement coating the red cell, monospecific testing is done. Now, agglutination with anti-IgG and negative reaction with anti-C3 indicates antibody or specifically IgG antibody alone is coating the cells. Agglutination with anti-C3 and negative reaction with anti-IgG indicates complement alone is coating the cells. But sometimes both IgG and complement may be present po. Now in the question, kanina, anti-IgG is negative but check cells are positive. That means the complement alone is coating the red cells. Next, for number 19. Let's go to microbiology. Okay, cerebrospinal fluid should be cultured immediately, but if delayed, the specimen should be. Again, cerebrospinal fluid, pa-highlight naman po ng CSF, it should be cultured immediately, but if delayed, the specimen should be. Choices. Letter A, frozen at -20 degrees Celsius. B, incubated at 37 degrees Celsius and cultured as possible. C, refrigerated at 4 to 6 degrees Celsius. D, stored at room temp for no longer than 24 hours. So the correct answer is letter B. Incubated at 37 degrees Celsius and cultured. Okay, so let's go to the specimen storage and specimen storage temperature for microbiology. So remember 4 degrees Celsius for urine, stool, also viral samples, and sputum. 37 degrees Celsius is used for CSF. -20 degrees Celsius for serum. And lastly, we have -70 degrees Celsius po for tissue samples and long-term storage po. Now, another thing for viral samples, transfer temperature is at -70 degrees Celsius po. But storage temp is 4 degrees Celsius. Yan. Next, for number, by the way, number 19, fastidious organisms po such as Neisseria and Haemophilus isolated from CSF of patients with bacterial meningitis are preserved by placing the fluid po in carbon dioxide at, again, 35 to 37 degrees Celsius po. Okay, or if the specimen could not be cultured immediately po. Number 20, furazolidone or furoxone susceptibility is a test used to differentiate. Again, furazolidone susceptibility is a test used to differentiate A. Staphylococcus from Micrococcus, B. Staphylococcus from Pseudomonas, choice letter C. Streptococcus from Micrococcus, or letter D. Streptococcus from Staphylococcus. The correct answer is letter A. Staphylococcus from Micrococcus. Now, Micrococcus and Staphylococcus can be differentiated by susceptibility to furazolidone. Remember, Staphylococcus is furazolidone susceptible or sensitive, and Micrococcus is furazolidone resistant. Number 20 po, letter A. Now, another one, just to add, to differentiate between Micrococcus and Staphylococcus, we also employ the OF, or oxidative fermentative test. So as you all know, OF test is used to determine if the bacteria metabolize carbohydrates oxidatively, fermentatively, or non-saccharolytic. When we say non-saccharolytic, that means the organism has no ability to use the carbohydrate in the medium. Now remember, oxidative organisms can only metabolize glucose under aerobic. Again, under aerobic conditions po, where the ultimate hydrogen acceptor is oxygen. No? Fermentative organisms, on the other hand, metabolize glucose, where the ultimate hydrogen acceptor is another substance. For example po, sulfur. So remember, Micrococcus po is oxidative, Staphylococcus is fermentative naman po. Okay. Next, for number 21, which of the following sugars is fermented by Staphylococcus? Again, which of the following sugars is fermented by Staphylococcus species? Okay, is it letter A, glucose, B, lactose, C, maltose, or D, sucrose? Okay, correct answer is letter A, glucose. So Staphylococci ferment glucose, whereas Micrococci fail to produce acid under, okay, anaerobic conditions. Okay, remember, under aerobic po, oxidative po si Micrococci po. For number 21, letter A. Next, for number 22, which of the following sugars is fermented by N-gonorrhoeae? Again, which of the following sugars is fermented by N-gonorrhoeae? Choices, letter A, is it glucose? Choice letter B, is it lactose? Or choice letter C, maltose? Or letter D, sucrose? Again, fermented by N-gonorrhoeae, correct answer is letter A, glucose po. In case you forgot, okay, N-gonorrhoeae, isa lang po, meron po siyang G. Tama? So, it will produce acid from glucose. For N-meningitidis, tama po ba? May M and may G po sila. So, N-meningitidis will produce acid from both glucose and maltose. We also have N-subflava. Take note, subflava letter S po. Okay, tatandaan nyo po, it will produce acid from glucose, maltose, and sucrose po. You also have N-lactamica po. N-lactamica, letter L po. Tama, lactose. Okay, so it will produce acid from glucose, maltose, and lactose. Also remember po, just in case, Moracella cataralis po fails to produce acid from glucose, maltose, sucrose, and even lactose. Next, for number 23, which of the following characteristics can differentiate Yersinia enterocolytica from Yersinia pestis? Again, which of the following characteristics can differentiate between Yersinia enterocolytica from Yersinia pestis? Okay, so choices. Letter A, motility at 25 degrees Celsius. Choice letter B, motility at 37 degrees Celsius. Choice letter C, fermentation of glucose, or D, hydrogen sulfide production po. Correct answer is letter A. Highlight naman po ng Yersinia enterocolytica at ng pestis. So remember, Yersinia species are non-motile po at 37 degrees Celsius. And all, lahat po ng Yersinia species po are motile at room temperature except kay Yersinia pestis. Because Yersinia pestis is again non-motile at room temperature. Now for number 24 po, which of the following Pseudomonas species is able to grow at 42 degrees Celsius? Again, which of the following Pseudomonas species is able to grow at 42 degrees Celsius? Is it letter A, P. aeruginosa, B. P. fluorescens, C. P. putida, or D. P. veroni? So the correct answer is letter A. Remember, P. aeruginosa, P. fluorescens, P. putida, P. veroni, and P. montelli comprise the group known as the fluorescent Pseudomonads po. Now, P. aeruginosa can be differentiated from the others in this group by its ability to grow po at 42 degrees Celsius po. With the exception again of P. aeruginosa po, again, the optimum temperature for growth of P. aeruginosa is 37 degrees Celsius, but it can grow to as high as 42 degrees Celsius po. Next, gram-negative straight rods associated with gastroenteritis po, with a biochemical test of in TSI A over A or K over AG and oxidase positive. Pa-highlight naman po ng oxidase positive. Is it letter A, E. coli, B. Yersinia enterocolitica, C. Citrobacter, or D. Aeromonas? Okay, correct answer po is letter B, Aeromonas. Again, pa-highlight po ng oxidase positive. For number 26, a 50-year-old non-immunized male with a persistent cough, fever, and flu-like symptoms was admitted to the hospital. Now, nasopharyngeal swabs were cultured on 15% blood, chocolate, Bordet Gengu, and Reagan low agars. Now, all media grew a gram-negative Coccobacillus. And also, carbohydrate and biochemical tests were negative. What is the most likely identification? Pilot naman po ng persistent cough. Okay, ano kaya yung answer sa choices na to? Is it A. Bordetella bronchoseptica, B. Bordetella pertussis, C. Haemophilus influenzae, or D. Haemophilus parainfluenzae po? Correct answer is letter B. Okay, Bordetella pertussis po. So, Bordetella pertussis is the cause of whooping cough, which is a highly contagious condition during the 5 to 10 day period after acquisition. Now, the incidence of whooping cough is greater in non-immunized individuals po. And therefore, it is higher sa mga bata under 1 year of age. Now, Bordetella bronchoseptica, on the other hand, is only rarely found in humans. Again, rarely found in humans, but it may cause respiratory disease in animals po. Now, unlike B. pertussis po, again, unlike B. pertussis, si Bordetella bronchoseptica po ay positive po for nitrite, urease, and motil po. Okay, so si Bordetella pertussis po kasi ay negative. For number 26, we have letter. Next, for number 27, which of the following is a rapid assay for Legionella species? Again, which of the following is a rapid assay for Legionella? Okay, choices is it A, antibody detection, B, culture on special media, C, direct examination on gram stain, or D, urine antigen detection po. Correct answer po is, okay, for number 27, we have letter D, urine antigen detection. Now remember, rapid assays po, useful for the detection of Legionella species are the urine antigen detection test plus DFA po. Pati DFA rin po, direct fluorescent antibody. Next, for number 28. Now, long, thin, gram-negative rods that are tapered or pointed at the ends. Again, highlight naman po ng tapered or pointed at the end. Choices, is it letter A, Bacteroides? Letter B, is it Fusobacterium? Choice letter C, is it Porphyromonas? Or choice letter D, Prevotella po. For number 28, again, highlight po ng tapered or pointed, we have letter B, Fusobacterium. Now, the term fusiform, which means long, thin, and tapered at the end po, is associated again with Fusobacterium po. So, you know, Fusobacterium species are, yan po yung mga characteristics nila. We have gram-negative, non-sporulating, non-motile, and they are filamentous po. And they are considered also as weak fermenters. Fusobacterium causes Lemierre syndrome po. Now, Fusobacterium produce N-butyric acid without isobutyric or isovaleric acid po. Okay, so this differentiate or distinguishes Fusobacterium from other Bacteroideaceae. Again, si Fusobacterium po ay nagpo-produce ng N-butyric acid po without isobutyric or isovaleric acid po.